Bismillahirrahmanirrahim. Now, as promised, I'm going to be putting in past papers nearly every day. So this is now a paper which is a paper 6-1, which is an alternative to practical, and this is the November 21 paper 6-1, and we're going to discuss that. Some of you might be taking this exam, some of you might not be taking this exam. So let me help those who are taking the exam. Now, as you all know, this paper is for 1 hour. And this is the paper 6-1, and this is the alternative to practical, and the total marks for this is 40. So the 40 marks for which you're going to be getting, you have to try to get as many marks as possible and finish this paper in 1 hour. Now, starting with question number 1, the tissue of a peeled potato tuber is composed of many similar cells. The student investigated the effect of varying concentrations of sucrose. Varying concentrations of sucrose. Immediately you should come to your mind 1% sucrose, 2% sucrose, 3% sucrose, and 4% sucrose. Just like you have a glass of water and you add 1 tablespoon of sugar in it, then you add 2, then you add 3. So different concentrations of sucrose solution on some potato tissue. She was provided with a potato tuber with the outer peel removed, distilled water labeled A, 3 sucrose solutions of different concentrations labeled B, C, and D. She cut 4 strips of potato tissue from the potato, each measuring 5 into 5 in cross section and 80 millimeter long. This was their length at the start of the investigation. Now, what I want you to remember is that whenever you're doing an ATP, think you're doing this in the lab. So the first part of the question was describe in detail how you would cut strips of potato to this size. Naturally, you would use a ruler, you would use a scalpel, you could have said a blade, you could have said a knife, and then of course you'd use a ruler, and you'd have a cutting surface like a white tile on which you placed it so that you could cut it very carefully. Then now let's look at the question now. I've drawn this drawing so make you all understand this is the way you should be thinking when you're doing this exam. The student then labeled 4 test tubes A, B, C, and D, so I've done that. She placed 1 potato strip in each and then added distilled water to the test tube A. So then she added distilled water to the test tube A. So we added distilled water. And basically, then they've said only distilled water to test tube A and the sucrose solution B, C, and D to test tubes B, C, and D. So B, C, and D contain maybe 1% sucrose, maybe 2% sucrose, maybe 3% sucrose. She made sure that each potato strip was fully covered by the solution. So here, remember, whenever we're talking of a liquid, we should specify the volume. Here, no volume is specified. So maybe this one has 5 ml water, this has 8 ml water, this has 9 ml. So this was something which you would need to pick up, that this was not very correctly, assessed, and we didn't say anything. We didn't say, okay, add 10 ml water, or maybe you said add 5 ml water, because then the water would be constant in all the test tubes. But nothing has been said. They just said she placed 1 potato strip in each, then added distilled water to test tube A. So distilled water was added here to test tube A, distilled water, and then B, C, and D, and the sucrose solutions B, C, and D to test tubes B, C, and D. She made sure that each potato strip was fully covered. Now, fully covered is not specifically in a volume. I mean, was this, maybe this had less water, maybe this had even lesser water and it was immersed in it. So maybe just if I draw it a little differently, then I can say, okay, this one was here. So there was less water in this. Maybe this one had more water in it. So something like this. So keep on thinking. After 30 minutes, so this is after 30 minutes, we left them in this liquid, whatever the liquid was. In A, it was distilled water. In B, it was sucrose. In C and D, it was sucrose again. She poured away the solution. So then we poured away this water. We threw away this water and prepared to remove the potato strips. So we're going to take a forceps and we're going to pick them up, because we shouldn't touch them, because that's not the way you do it in the lab. So you took a forceps and you pick them out. So remove the potato from the test tube, and then she put it against a ruler and she measured them. Then it says describe what she could do to ensure that after they had been removed from the test tube, she knew which potato strips had been in which solution. But if she just takes them out and just place them, she needs to then do something to mark it. Okay, this one is the one, this is the one from tube A, which was distilled water. This is the one from B, which was 1% sucrose. This is the one from C, which was 2%. And this is the one from D, which was 3% sucrose. Now that was very easy because all she had to do was that she had to either label petri dishes or beakers and label them A, B, C, D. So then if she labeled them A, B, C, D, then she would place the A into A and the B into B and the C into D. Or she could have labeled a white tile and then she could have placed the pieces. And so on a white tile, she wrote A, B, C, D, and then from wherever she took out it, she would place it on that white tile. So she would know that this potato strip was from the beaker A and the other was from beaker B and C and D and so on. Then we have the diagram shows the four potato strips after 30 minutes in the solution. So we have A, B, C, D. Then you measure the lengths of strips A, B, C, and D and record the lengths of each strip at the start and end of the investigation and calculate the change in length. Enter your results in the table. So you have to just use a ruler and you have to measure this and then you have to talk about that. So now when we look at it, all the initial lengths were 80. But A went up to 85, so that's a plus 5 change in length, and then 80 to 79, so that's a minus 1. Then 80 to 75 is a minus 5, and then 80 to 82 is a plus 2. Then flexibility of the strip we didn't know because we hadn't done this practical, so we just were going to look at the question. So then it says she also assessed the flexibility of each strip by gently bending them. So she just tried to bend them a little, and then more easily the strip bent, the more flexible it was. She recorded the results in a notebook. Strip C was most flexible. Strip A was least flexible, and strip D was more flexible than A but less than C. And strip B more flexible than D but less flexible than C. So this was easy to do because strip C was most flexible. So as we look at the next part, they gave you these figures. They said, okay, first you do this, arrange the strips in order of the decreasing flexibility and use the scale below to record this in the table. Record the most flexible as plus four signs, and the least flexible as 1 plus. So this was 2 marks. So you had to do this on the table previously, and I have done this here. So C was the most flexible, so that would be 4, and A was the least flexible, so that would be the 1 plus, and this was the more, and then you had to figure it out. Strip D was more than A, so D was more than A, so that will be 2 plus, but less than C. So C was 4 plus as it is, and then it says B was more flexible than D. So B was more than D, so it had to be more than D, so it had to be this 2 and this had to be 3. So you had to figure this out, not very difficult. I'm sure you could have done it. Wasn't very challenging at all. Now coming to part 5 of the question, describe and explain the changes in length and flexibility in the potato strip that had been in solution A. Now if you look at solution A, this is solution A. It had increased in length the most. It was 85 length and the flexibility was least. Now it says describe and explain. So when it says describe and explain, well, then you have to give the description that the strip was longer, is the longest in fact, and it was the least flexible. Why? Because in water, the water moved in by osmosis and the cells became more turgid, or there was an increase in turgor pressure. So they became so it was least flexible. You couldn't bend it easily, just like you fill a balloon with air and you can't bend it. But if you've got less air in it, then of course it's more flexible. So this is what you had to give it, that the strip was longer, least flexible, water moved in by osmosis, and the cells became more turgid, or there was increase in turgor pressure. So this is how we would have written it. Strips longer, least flexible, water moved in by osmosis, and cells more turgid or increase in turgor pressure. Now coming to part 5, it says identify 2 problems with the method used in this investigation that might have led to unreliable conclusions. Suggest a way in which could have been improved to make the results more reliable. Now, these are very favorite questions in the alternative to practical exam, and you must know how to handle these and very correctly. So you look at the practical and you think, okay, what could I have done wrong in this? So, I mean, the first point which comes to your mind is maybe it was not exactly 80 millimeter. So it is difficult to cut to the correct size. So either you use a very sharp or a finer blade, or you use a corer, or you use something which makes the chip making machine. So what was the problem and what was the improvement? So you have to talk about the problem. What was the problem you faced and what was the improvement? You can only figure this out if actually you think you're doing this in the lab, and then you visualize yourself and you sort of do a simulation, and then you think, okay, this could have been going wrong. So the first two ones which I'm discussing with you all is difficult to cut the correct length. So the improvement to that was use a finer blade. Then the second problem which one could have said was you used only one strip. Now that is never a thing. We always use more strips, or we repeat the experiment so that the results become more reliable. Because if you used one strip only, maybe that strip was a damaged piece of the potato, maybe that potato was diseased potato, or that strip had some diseased cells, so that is not a very correct way of doing it. Like I say, okay, well, I'm just going to decide your monthly grade on one test. But if I give you three tests and maybe then I take out the average, and one test you didn't do so well, and then the other one you did better, and the third one was okay, so then we would take out an average, and we would not base our remarks or our grade on just one test. So if I make you do it three times, then you get a better average, and it becomes more reliable that, okay, this student has this caliber, so it gets these marks in the exam. So difficulty to cut the correct length, and then use a finer blade, or you use a cork borer, a corer. You can also use this, or you can also use uh, apple corer, which is used to remove the inside of the that's called a apple corer. I'll just show you a diagram of that. Then only one strip was used, so more strips. Then if you look at another way that we could have said is that not much change in length. So if we leave them for a more longer in this more if we take more longer strips, then we could probably get a better change in length, would be able to be figured out. Then the other which we could have done is time in the solution was different. Now, what does this mean? Time in the solution was different. I'm sure most of you do not understand what this means. Well, actually, when you took the four test tubes and then you said, okay, put the potato in this. So you put the potato, say, at 10:00. Then you put it at 10:01. And then you put it the one in 10:02. So 1 minute, 10 o'clock, 10. So then you were actually putting them after some time. So it was 10:01, then 10:02, then 10:03. So you were actually putting it after some time. And then you said leave them for 30 minutes. So the one in the end was for 30 minutes, but this was for 31, this was for 32 minutes, and this one was for 33 minutes. So understand that, what do I mean? That time in solution is different, because when you placed it in, say, test tube A, then after a minute, you took a minute to then take another piece and put it in test tube B, and then put it in test tube C, and then put it in test tube C. So you took and then you said, okay, now I put them in all the four test tubes. Now let's time it for 30 minutes. So in some solutions it was 31 minutes, in some solutions it was 32, in some it was 33 minutes. So time and solutions difference, time each strip separately. So you put it in one and then wait for 30 minutes and then do it. Or the other point, which was this was flexibility, was very subjective. I mean, you said less flexible, more flexible. I mean, we didn't have any meter or any thermometer or any gauge by which we measured it. So it was all very very subjective, or it was difficult to assess. And some you said less flexible or more flexible, or this was more than this, or this was all very very vague. And that's purposely they leave that so that you can pick that up. So then it says hang masses on the strip. Now you realize that the strip, if you have masses on the strip, then it'll bend, and then you can see the curvature of the bend. Or you can say leave for the longer time. Now I'm just going to show you some diagrams about this. This is a diagram which shows how we would do this and how we would place this. Of course, this is a much more crude way of doing it, but we would do it in the lab in a proper with test tubes. The diagram which shows this in a slightly better manner. Another diagram for you all to see how we would use this to really take out the potato strips. Now, coming to the B part of the question, another student investigated the effect of varying concentrations of sucrose solutions on some potato tissue by measuring changes in the mass of the tissues. You see, at times the length might not have increased, but the breadth might have increased, and you think, oh my God, length hasn't increased. So mass was a much better way of doing it. That's always a very better sort of improvisation on the previous experiment. He placed 5 pieces of potato tissue, each with a mass of 5 grams, in 5 separate test tubes. The test tube contained either distilled water or one of the 4 different concentrations of sucrose solution. So imagine 4 different concentrations of sucrose: 1%, 2%, 3%, and 4%. You must have developed this skill in O levels. Later, he poured away the solutions. State two factors apart from the mass of the tissue that should have been kept constant when using this method. Naturally, what should have been kept constant was the temperature, the volume of the solutions, the time of the pieces of potato in the solution, and the same potato and the surface area of the potato. So temperature, volume of the solution, time, piece of the potato in the solution, the same potato should have been used because genetically it should be the same. You couldn't have used two or three different potatoes and they would be genetically the same, different potatoes. And the last is the surface area of the potato. When he removed the pieces of tissue from the test tubes, he carefully dried them on a paper towel. Explain why it was important to dry them. Why would we need to dry the potatoes? Naturally, of course, the main thing is remove the excess water and only want to measure the mass of the tissue. You don't want to measure the sucrose solution which was on the surface of it, which might have been added maybe a few micrograms or whatever, but it would change the mass. So remove excess or the surface water. You must be very correct in wording this because excess water is a underlined mark scheme point. So that means this word had to be used. Remove excess water or remove the surface water. And only the mass of the potato should be weighed, not whatever is coating it as well. Then we come to the next part. The table shows the measurements at the end of the investigation. Sucrose concentration in mole per dm cube. You always write the units in the heading, and this is, of course, always going to go on the x-axis. The first column, this is the first column, always goes on the x-axis, and this is why we call it fixed. First column is the independent variable, and it always goes on the x-axis. This is what you're going to remember in your A levels as well. And then the mass at the end of the investigation, this will always go on the y-axis. This is a rule that we follow. Please remember that. So mass at the end of the investigation is this. Construct a line graph of the data in the table on the grid below. Join your points with ruled straight lines. Okay, so then you have to divide this graph. Please remember you have to look at the smallest figure and the largest figure. So this was 0.0 and 0.8. So 0.8 must come somewhere here. While on the y-axis, so 0 must come here and 0.8 must come somewhere here. And on the y-axis, we have the least figure is 3.3 and then we have 5.7. So you've got to see is that you've got to spread the data here. So this should be somewhere here. We should start with 3 and then we should have 6 somewhere here. So this is how you would divide it because the values are between 3.3 and 5.7. First of all, you must label the axis and you must write the entire story. This entire story must be written here. Sucrose concentration slash mole per dm cube. So you wrote that, you actually get a mark for labeling the axis. And the y-axis, you have to write the whole thing, mass at end of investigation and slash grams. Units must be written. You must write the units because if you don't write the units, well, you lose a mark for that. So if you label the axis, it says sucrose on x-axis and fully labeled with units, you got 1 mark. And then linear scale and origin labeled and good use of the grid. So you spread the grid in such a way that you have to spread it. I spread it like this. You could do it differently if you wanted it, but it should not be some weird like 7 squares for something. It should be something very, very standard, which we do all the time in biology. In A levels, of course, the scale is in the mark scheme. The scale must be according to the mark scheme, not here in O levels. We don't really looked into that a lot. So then you need to, now you've done this, you've labeled the axis. Now there are 2 marks you get if you haven't done anything on the graph and you've just labeled it and you've decided the scale. Well, you've got 2 marks now. But then when you plot it's another third mark and then you connect it correctly, and then that's the fourth mark. So 4 marks for the graph are for these 4 points. Then you plot the graph and you see there is a 0.5 in it. It's not very regular. It's 0.0, 0.2, 0.4, and 0.5 and 0.8. But this way you could have made a mistake. I nearly made that mistake too. So you plotted the points. Now you still get another mark for it if it is correctly plotted. And then the next mark is for joining it. Join it with a ruler, point to point with a ruler. That's a very good way. And the plotting can either be this or you can do it with an axe. Please do not mix the two. So if you're just going to do the dot and the dot and the then you just do the dots, but you have to circle the dots. So please remember you have to circle the dots, and then you do with the ruler, you connect the point to point. And that of course is the best way to do it. If you're doing it across, then of course you point and take the center of it and you join it. So please remember, joining it is a very important, and you can't lose this mark. You must get these 4 marks of the graph. These are very easy marks to get. Then the next part of the question is part 4. Use your graph to determine the concentration of sucrose, which will produce no change in mass. Now, if you look at the original, what was the mass? The mass was 5 grams. So you have got to go back to the 5 gram story. So then use your mass, and this of course was as given in the mark scheme, it is 0.27. 0.27 was the sucrose. Use your graph to estimate the concentration of sucrose. So 0.27, but then you had to give me the mole per dm cube story as well, because if you didn't give me that, you didn't get the 2 marks. The 2 marks are for what? One is for the units. So 0.27 mole per dm cube. But how did you figure this out? Describe the method you used to determine the concentration of sucrose. Well, it was very simple. You had to go to the 5 gram. Now you go on to the 5 gram of the graph. So this is here, you would have gone on to this. And you would just make a line here where it touches it, and then you would read it off on the x-axis. This is where you would read it. Why? Because 5 gram means no change in mass. Was 5 grams was the original mass. So we used 5 grams. So describe the method you could determine the concentration of sucrose. Was the original mass of 5 grams, and as point to interpolate line from the y-axis. Now coming to question number 2, the photo micrograph shows the central part of a plant root as seen under a light microscope, and we have cells labeled E, G, and F. Then we say the xylem and phloem are surrounded by a layer of tissue that is a 1 cell thick. The cell labeled E is in this layer. So let's look at cell labeled E. So this is cell labeled E, and this is the cell that we are talking about here, right? The cell labeled E in this layer, look at it carefully. Then it says make a large drawing of cell E and the cells that touch it on the outside and inside of the layer. So we're going to be drawing this area, outside and inside. So I would say we draw this one, and this one, and this one. So it says drawing E and the cells that touch it on the outside and inside of the layer. So this should be as big as 5 centimeter, outline of the cell E drawn with a sharp pencil and no shading anywhere, cell E with inner and lateral walls thick. Then cell E only 2 cells on the inside and 2 cells on the outside, and both outside cells larger than the inside cells. So I'll just do a diagram of this to show you, and you just have to do it. You just have to draw those cells, and I'm sure all of you could have got your, because this is 4 marks and these are very basic drawings, and they're not, we don't expect you to be an artist or something, but you need to do it really carefully. Now you can see the diagram. It's a very simple diagram. I'm not being able to do it with my marking with my pen that I have on my using on my iPad, but I have to use double lines to show the thickness of the cell wall, but the 2 lines must be very close to each other. They have to be that's very, very close to each other. Just see them as 2 lines very close to each other. And then you had to draw the cell E. So 5 centimeter was the minimum size. So you must cover this area. You see, you must remember this area given to me where the mark is. You must cover 2/3 of this area. So 5 centimeter was the minimum size. Of course, you could have drawn it bigger. And outline of cell E drawn with a sharp pencil and no shading anywhere. You can see as I've done this, this is cell E. And then I've shown it that cell E with a thick inner and lateral wall. So this inner and lateral walls were thick. So I've just drawn this line to show that. This, of course, I've tried to do a double line showing the cell wall, but I've not been very good at that. Then cell E plus only 2 cells on the inside. So these are the 2 cells on the inside. These are the 2 cells on the inside, which I've drawn, and 2 cells on the outside, and both outside cells larger than the inside cells. So this is what you had to do, and this is how you have to draw it. So not very difficult, but you need to read the question carefully and do it as best as you can. Then coming to part 2, measure the diameter of the xylem vessel between F and G. That was 17 millimeter. You always use the units. If it wasn't given, you would get a mark for that. This would have been 2 marks. Always use the unit millimeter. I've always asked you to do that. Calculate the actual diameter. Why are we dividing it by 200? Because you can look in the diagram. The diagram has a magnification of 200. So please look at the magnification in your question. It might be 400, might be something else. So you divide it by the magnification and you get a value of 0.085 millimeter. So you need to be very careful in doing it to a number of decimal points, that is 3 decimal points here. Then it says some of the outer cells of the photomicrograph contain small particles. Describe in detail how you would prepare a section of the root for observation under the microscope. Now this again syllabus point hai. This is one of the syllabus points that you need to know how to examine under the microscope. So you cut a very thin slice of the root, you place on a microscope slide, you stain the section with iodine. Why? Because they've said you have to contain starch. So you have got to think of how would you stain the starch. You stain the section with iodine, add a cover place, you place it at an angle so you avoid bubbles. You place the slide on the microscope and focus the lens and put the light on, and use stains black if starch is present. So thin slice, microscope slide, iodine, cover slip. This is for every microscope question you can write all that, and stains black if starch is present. That completes this paper, and I wish you all the best, and please do subscribe, and please do tell your friends about the channel, and please let me know if there are any comments in which I can help you in any other way. Thank you very much.